Good morning, everyone.
Nearby two device.
Sir, can you hear me?
Yes, ma'am. One of the device must be muted, ma'am. I think
Is it fine now, sir? Is it echoing? Winston, sir?
you can start now.
Hello?
Madam?
Okay, thank you, sir.
Everything is okay. You
A
can start
very
now.
happy morning and a warm welcome, sir. A very blessing team to the first day of the 3-day national level zoology webinar series 2020. We pray and wish that this webinar will help us enhance our understanding of our present knowledge on various and inspire and open new research ventures to the young minds. I now request our Father Principal, Reverend Dr. S. Mariadas S.J., to offer his felicitations and blessings.
Okay. Good morning, everybody. Are you able to hear me?
Yes, Father.
Kekda.
Yes, Father.
Okay. This is Father Mariadas, the principal. Good morning once again to everybody. I'm very happy to address all of you on this wonderful day. I appreciate the HOD and all the professors of zoology department for organizing this national webinar on a particular topic. I'm very happy to note that so many people have joined to enrich themselves academically during this difficult time. Though I like to speak a lot, I'm busy with another important meeting. I have come out of the meeting. That's why I thought I should go back immediately. Therefore, I once again appreciate the HOD and all the professors for wonderfully taking this particular webinar. I also welcome all the participants on behalf of St. Xavier's College. As many of the participants from our college know very well, St. Xavier's was established in the year 1923, and very soon, in another 2 or 3 years' time, it will celebrate its 100 years of its academic existence in this part of Tamil Nadu and in this part of country. And when I think of this zoology department, this is a particular department which has contributed a lot to the society over these years. Academically, it has contributed a lot. Definitely, I know the history of, let us say, the zoology department from the year 1984 onwards. We had a number of stalwarts who have gone out of their way to contribute academically to this college and to the society. A number of our alumni who live elsewhere, they still even now remember very gratefully our institution for instilling value education from this institution. They greatly remember all the staff members who have molded them, who have shaped them. Therefore, once again, I register my total sense of gratitude to all the professors for organizing this. I wish you all the best. Have a wonderful day. Thank you.
Thank you very much, Father. Now I request our head, Department of Zoology and convener of this webinar to welcome and highlight the theme of this webinar.
Respected Reverend Fathers, respected speakers of the day, respected IQAC coordinator, respected faculty members of Department of Zoology, participants from various colleges and universities, and my beloved students, a warm good morning to each and every one of you. On behalf of the Department of Zoology, I am happy to welcome you all to the National Level Zoology Webinar Series being organized between 10 a.m. and 12 p.m. on 22nd, 23rd, and 25th of July by the Department of Zoology, St. Xavier's College, Palayamkotte, Tamil Nadu. At the outset, I welcome Reverend Father Reverend Dr. V. Henry Jerome, the Rector of Sovereign Institutions. He is an eminent educationist and a renowned social reformer. Welcome, Father. I express my cordial welcome to Reverend Dr. Alphonse Manikam, Secretary of St. Xavier's College, an economist and a nature enthusiast whose indomitable leadership is reflected in all the developmental activities of our college. Welcome, Father. It gives me an immense pleasure to extend a warm welcome to Reverend Dr. S. Mariadas, Principal of St. Xavier's College, a man of dignity and strong determination who has been successfully leading our college in the right direction during this pandemic storm. Welcome, Father. On behalf of all the participants connected online, I welcome today's resource person, Dr. Hubert RS and Dr. V.J. Jins. Welcome, sir. This webinar highlights the topic more relevant to these days, emerging zoonotic viral diseases, innovative strategies in the development of antiviral drugs, bioprospecting of marine bioresources for pharmaceutical, present status of Western Ghats biodiversity, and a new insights on cnidarians. I welcome to kindly join all the lectures. Main objectives of this webinar are to appreciate the species richness, variety, and a wide range of life that forms our unique biosphere, to engage ourselves in continuous learning process during the pandemic crisis, and finally inspire and ignite the young minds to become socially and environmentally conscious so that they will be able to find solutions for the emerging problems from a scientific perspective. I once again welcome the participants from different colleges and universities across India. I welcome and thank our college technical team for their support throughout the webinar. Happy learning. Thank you, one and all.
Thank you, Raja, for your kind words of welcome. For your kind words of welcome. Let us now move into the first session by Dr. Hubert Darius J. Daniel. I'm happy to introduce Dr. Hubert to you all. Dr. Hubert Darius J. Daniel did his BSc Zoology in Madras University, MSc Microbiology in Periyar University, PhD in Christian Medical College, and he is the postdoctoral fellow at the Department of Pathology, Johns Hopkins School of Medicine. His professional experience includes being an associate research officer in Christian Medical College and a research trainee in Johns Hopkins School of Medicine. He is awarded IIMM Gold Medal for Best Published Paper in Virology for the years 2006 and 2009 by National Congress of Indian Association of Medical Microbiologists. He has published 28 peer-reviewed original scientific research papers, presented in 6 international conferences, and has completed 4 research projects. His research area includes molecular, immunological, and epidemiological studies of hepatitis infection, writing standard operating procedures.
I'm sorry for the technical difficulty. Feeling honored to have Dr. Hubert Darius J. Daniel with us today, sir. You may share the screen and begin your presentation. Kindly take over the session, sir. And for the participants, you can post your questions in the chat window and your questions will be answered towards the end of the session. Dr. Hubert?
Thank you, Dr. Mabel. Thank you for the organization for inviting me to give this talk on zoonotic viral infections. Zoonosis is defined as the disease and infection naturally transmitted between vertebrate animals and humans by WHO. According to WHO, zoonosis in Southeast Asia are categorized into endemic, re-emerging, emerging with epidemic potential. The zoonosis is classified into bacterial, viral, rickettsial, protozoal, helminthic, fungal, and ectoparasites according to its etiology. They are further classified based on the mode of transmission into direct zoonosis, where the infected vertebrate host infects directly to the human, like in rabies, cyclozoonosis, where it requires more than one vertebrate host, but there is no invertebrate host for the completion of its life cycle, metazoonosis, where it's transmitted biologically by an invertebrate vector in which some part of development happens, and saprozoonosis, where it requires the vertebrate host and a non-animal development site such as soil. It is also divided into anthroponosis, which is transmission of the virus from humans to lower animals, zooanthroponosis, when it's transmitted from human to lower animals, and amphizoonosis, when its infection is maintained in humans and the animals and is also transmitted both ways. Out of the 1,415 microbial diseases, 61% or 863 of them are zoonotic. And of the 177 emerging viruses, emerging infections, 13% or 75% of that is zoonosis. The major problem with zoonosis is because it spreads very fast when it's an emerging infection, and because of the global connectivity, it can spread very quickly, and it causes huge economic burden, especially to animal trade and travel. The major drivers of emerging and re-emerging zoonosis is the changes in the land use or agricultural practices where we acquire the forest, changes in human demographics, poor public health, hospital and medical procedures, pathogen evolution, mutations, contamination of food source or water supplies, international travel, failure in the public health system, international trade, and climate change in the order of significance. The zoonotic infections are of major concern because new viral pathogens continue to emerge, and it affects global health as they have a potential to be an epidemic and cause high case fatality because of absence of specific treatment, and non-availability of vaccines to control the spread, which again causes a huge economic burden as we see with COVID-19. These are a list of important viruses which are zoonotic. Rabies virus, which is a Lysa virus. It's transmitted by dogs. Japanese encephalitis virus, which is a flavivirus, causes encephalitis and is found in pigs and wading birds. Dengue virus, which is again a flavivirus, causes dengue fever, dengue hemorrhagic fever, and dengue shock syndrome, found in monkeys. Nipah virus, which is a henipavirus, which causes respiratory to fatal encephalitis. Casanova forest disease, which is found in a Casanova forest and causes Casanova forest disease. It's a flavivirus. Chikungunya virus, which is a toga viridae, causes chikungunya disease. West Nile virus, which is again a flavivirus, causes West Nile fever. Zika virus, a flavivirus again, it causes microcephaly in children and Guillain-Barré syndrome in adults. Yellow fever virus causes yellow fever, which is again a flavivirus. Ebola and Marburg, which belongs to Filoviridae, causes Ebola hemorrhagic fever and Marburg hemorrhagic fever. Lassa virus, which is an arena virus, causes Lassa fever, and Rift Valley fever, which is a flibovirus, causes Rift Valley fever. And Crimean Congo hemorrhagic fever causes Crimean Congo hemorrhagic fever, which is a Bunia viridae. And avian and swine flu causes flu, which is found in swine and birds, which belongs to Orthomyxoviridae. And finally, SARS-CoV coronavirus, MERS coronavirus, and SARS coronavirus 2, they belong to coronavirus, causes respiratory syndrome. SARS coronavirus, which is found in civet cats, which is transmitted from bats to civet cats to humans. MERS coronavirus, which was found in camels in the Middle East, and SARS coronavirus 2, which is most probably from bats. Rabies virus, which belongs to the family Rhabdoviridae and the genus Lysa virus. It's a bullet-shaped virus which is enveloped and has spikes around it. It's an RNA virus. As you would notice here, all the viruses or most of the zoonotic viruses are RNA viruses since they mutate very fast. Rabies is estimated to cause around 59,000 human deaths annually in about 150 countries, with 99% of the rabies being caused by dog, and 96% mortality and morbidity is associated with dog bites. Cat, wolf, jackal, mongoose, and monkeys are other important reservoirs which are found in India. This map shows us the death rate found in different countries, with India rating very high, the dark brown, and it's about 12,700 deaths per year. Rabies virus is inoculated when a dog bites, and the virus replicates in the muzzle, and from there it spreads to the through the nicotine acetylcholine receptor to the neuromuscular junction, where it enters the axons and then to the peripheral nerves, and is transported to the spinal cord, from where it replicates in the motor neurons of the spinal cord and then enters the brain. From the brain, it travels to the eye and the salivary glands. The symptoms include fever, headache, excessive salivation, muscle spasm, paralysis, and hydrophobia as the pathognomic feature. In dogs, it causes change of behavior, bark tone, feeding habits, excessive salivation. In cats, it causes extreme aggression and may attack dogs or man. In cattle, it causes abnormal movement of the posterior extremity, with foamy yellow froth from mouth. No hydrophobia is not observed in animals. Lab diagnosis is done by collecting saliva, sputum, corneal smear, skin biopsy, CSF, blood, urine, and brain tissue. Negri body is identified in the brain tissue by histopathological staining. Antibody detection is done by indirect fluorescent antibody test, serum virus neutralization test, counter immunoelectrophoresis, and ELISA. Electron microscopy is used for direct identification of the virus, and corneal smear is also done. Molecular diagnosis is done by RT-PCR. There are different types of vaccines which were developed. One in human diploid cell vaccine, purified vero cell vaccine, and purified chick embryo cell vaccine. Purified vero cell vaccine has good safety and high immunogenicity. These are the WHO guidelines for post-prophylaxis. It's classified into three: touching and feeding of animals or licking. You don't have to treat them. In the second category, where there's a nibbling of our skin where there is minor abrasion, then a vaccine is administered and the animal is watched or observed for about 10 days. And otherwise, the animal can be killed and has to be proved that it does not have rabies, then the vaccine can be stopped. When there is a bite by the animal, then immunoglobulin, rabies immunoglobulin has to be administered around the site, and vaccine also has to be administered unless the animal is proved to be healthy. Post-exposure prophylaxis, the first prophylaxis to be done is wound care, and then passive immunization and active immunization. Wound care is thorough washing of the wound with soap and water, and then with povidone iodine to clean. And tetanus prophylaxis and antibiotic has to be given depending on the type of wound or the deepness. And rabies immunoglobulin has to be administered immediately at the site of the wound, and on the opposite arm, the vaccine has to be administered. And the vaccine given as human diploid cell vaccine or the purified chick embryo cell vaccine, which is administered on 0, 3, 7, 14 days. And if the person's vaccinated before, then immunoglobulin does not have to be administered. Pre-exposure prophylaxis is given for veterinarians, veterinary students, or veterinary technicians who have high exposure or work closely with animals, and lab workers working with rabies virus or material specimens that might have the virus. Other person whose vocation bring them to contact with animals are also at risk. They're classified into three groups: continuous risk, frequent, and infrequent. Continuous risk, people has to be tested every 6 months, and frequent every 2 years, and infrequent do not have to be tested. The protective level of antibody is 1 in 5 serum dilution by rapid fluorescent focus inhibition test. Rabies vaccine is, as a primary dose, is given on 0, 7, 21, or 28 days. And if the neutralization antibody is not present or is greater than 1.5 serum dilution, then a booster is given. Japanese encephalitis virus is an inapparent acute arboviral infection, which is mainly found in horses, pigs, and humans. Belongs to the Flaviviridae. and it's a spherical lipoprotein enveloped protein with spikes, and it has a single-stranded positive sense RNA. This is the geographical spread of Japanese encephalitis, found mostly in China, Cambodia, Indonesia, Japan, and it was first reported in Vellore and Puducherry in the mid-1950s, and there were outbreaks in 1973, 76, and 78 thereafter too. The worst outbreak was in 1988 in UP. It caused 1,413 deaths. And between 2010 to 17, there were reports of 222 deaths per year. Each outbreak coincides with the monsoon and post-monsoon period when the vector density is high. The case fatality rate in newly affected areas range from 10 to 70%. This is a life cycle of JE virus where the amplifying host is the pig and the birds. They are transmitted by mosquitoes, and man is a dead-end host from where it's not transmitted back into the system. The signs and symptoms, the incubation period is 5 to 15 days with a course of disease in about 3 stages, which is prodromal stage, acute encephalitis stage, and the late stage. In the initial prodromal stage, you have fever, headache, and malaise. Acute encephalitis stage, you have fever and focal CNS signs and convulsions. Lab diagnosis is done indirectly by antibody titer, where IgM, JE specific antibodies are detected in serum or CSF. And specific diagnosis is by virus isolation from CSF or brain. No specific treatment is available. Supportive care is given. Preventive measures are controlling the mosquito breeding and JE vaccination. And it's recommended only for use in endemic area. Dengue virus belongs to the Flaviviridae family, and it's a single-stranded RNA virus. It has 4 serotypes, dengue 1, 2, 3, and 4. The transmission of, the control of dengue virus is complicated because it is maintained in the mosquito by transovarian transmission. Dengue 2 and 1 is found in North India. Dengue 2 and 3 is found in South India. Northeast region has dengue 3, 1, and 2. In India, the first outbreak was reported in 1812, and then in 1996 there was a large outbreak of dengue fever and dengue hemorrhagic fever around the Delhi region, dengue hemorrhagic fever and dengue fever in 2003, and thereafter too. In 2017, there was about 1,57,000 cases reported, mostly from Tamil Nadu and Bengal. This is the geographic distribution of dengue, which is found along the tropics, with India having quite a few big dots, about 1 million cases. The incubation period for dengue is 5 to 6 days, and dengue fever is an abrupt onset of fever with maculopapular rash, muscle joint pain called breakbone fever, retroorbital pain, nausea, and vomiting. Dengue hemorrhagic fever is a high-grade fever with hepatomegaly, thrombocytopenia, and a raised hematocrit. Dengue shock syndrome is dengue hemorrhagic fever plus a shock. Transmission of dengue happens through mosquito, and when a mosquito bites, the saliva transmits the virus, and the virus localizes at the site and replicates the target organs, which is lymph node and liver. Virus is released into the other organs through blood, and it infects WBCs and other lymphatic tissues. Virus is released and circulates in the blood, and the mosquito ingests the blood, which is transmitted back. to others. In the mosquito, the virus replicates in the mosquito midgut, ovaries, and nerve tissues, and it goes back to the salivary gland from which it's transmitted again. Lab diagnosis is done by serum, plasma, or whole blood. Dengue virus is isolated using mosquito cell lines. NS1 antigen, which is found during the early onset of infection, is detected by ELISA or a rapid method. Antibody detection is done by IgM ELISA, IgG ELISA, or rapid test kits. Molecular detection is done by RT-PCR. Vaccine, there's a vaccine which is licensed to use from 2015. It's a live attenuated chimeric yellow fever dengue tetravalent dengue vaccine, which is commercially called Dengvaxie. It's approved in Mexico, Philippines, Brazil, Indonesia, Thailand, but it's not approved in India, but India is developing a vaccine against dengue. And it's administered for people from 9 to 45 years old. Prevention is by personal protection against mosquito, environmental protection, reducing the vector breeding site, public education, chemical application of pyrethron and malathion, and biological control using larvivorous fish, which feeds on the larvae. Nipah virus, which was first recognized in 1999 in Malaysia, and is also found in Kerala, is a non-segmented single-stranded RNA virus which belongs to the Paramyxoviridae family and the genus Henipavirus. The natural reservoir is the fruit bats, and it is transmitted by humans consuming the partially eaten fruits which are consumed by the bats. Its transmission is direct contact with the infected bat or pigs, and it's transmitted by the body fluids which are contaminated by the body fluids of the bat which are infected. Case fatality for this virus is pretty high, 40 to 75%. It is transmitted by large droplets by inhalation or by aerosol. It infects the lung and causes respiratory syndrome. It also infects the cranial nerve and goes to the brain, causing encephalitis. Incubation period is 4 days to 2 months, and patient presents with fever, headache, dizziness, and vomiting, severe encephalitis, nosocomial. Nosocomial infection is also very common. The preferred samples are throat swab, urine, or blood, and CSF. Lab diagnosis is done by ELISA, serum neutralization test, PCR, virus isolation in vero cells, and electron microscopy. Prevention is by controlling the consumption of contaminated sap, or covering the pots which are used to collect the contaminated sap, used to collect the sap from the trees, and good surveillance and awareness and reliable lab assays and standard precautions have to be done to avoid nosocomial infection. There are many vaccines which are developed, which are on the course. Kiasnur forest disease is a disease which is transmitted from monkeys or infected cattle, and it's transmitted directly to humans It's found in the Kyasnur forest region of Karnataka. It's found mostly in the Chikmagalur, Udupi region of Karnataka state. There was a large outbreak in 83-84, and it's a very important emerging, pre-emerging viral infection. In 2013, it was detected in dead monkeys and Nilgiris in Tamil Nadu, and also in Kerala. KFT outbreaks have been reported in North Goa. The incubation period is 3 to 8 days with an acute and a second phase. In the acute phase, there's fever, headache, myalgia, dehydration, nasal cavity, throat gums, GI disturbances, and hemorrhages. In the second stage, goes on to cause the meningoencephalitis and can occur after 21 days. Lab diagnosis is done by virus isolation from blood and IgM antibody detection by ELISA. The molecular detection is done by RT-PCR. Prevention is controlling the vector, which is the tick, by using repellents or spray, and a formalin inactivated chick embryo vaccine, which is developed by Hafkin Institute in Mumbai. Is recommended for the endemic areas. Chikungunya is a self-limiting virus, which is described as an outbreak in South Tanzania in 1952. Chikungunya belongs to the Tobaciviridae family and the alpha virus, which is transmitted by Aedes albopictus and aegypticus. Reservoirs are monkey, birds, and other vertebrates. Transmission is by bites of the female mosquito to humans. The virus multiplies at the site of infection, at the site or port of entry, and viremia happens there, and a good antiviral immunity is also created. The main target cells are the keratinocytes and the melanocytes. It causes arthralgia. It causes permanent or arthritis as well. As a complication, it infects the organ and causes neurocognitive impairment. The incubation period is 2 to 12 days, and the symptoms are headache, nausea, fatigue, and it can be misdiagnosed with dengue and Zika. The samples are serum or plasma, and viral nucleic acid can be detected. IgM, ELISA, and neutralizing antibodies are also detected in the early stages of infection. Virus culture can be done in the first initial 3 days of illness, and RNA can be detected for the first 8 days of illness. And IgM antibody is found in the first week up to 14 days of illness. No vaccines are available. Preventive efforts are focused on vector control and elimination of breeding grounds using larvivorous fish, which feeds on the mosquito larvae, and insect repellents are used. Good epidemiological surveillance of chikungunya has to be done with a good community education for transmission control. West Nile virus belongs to the flavivirus genus and Flaviviridae, causes neurological disease in humans. Approximately 80% of the people are asymptomatic. It's transmitted through the bites of infected mosquitoes in humans, horses, and other mammals. Birds are the natural host of West Nile virus. West Nile virus is commonly found in Africa, Europe. It was first identified in a woman in West Nile district, hence the name. And during a virulent epidemic in 1997, caused encephalitis and paralysis in birds. It was widely established in Canada to Venezuela. The mode of transmission is through the mosquitoes to humans and to animals. It's usually found in the birds. It's transmitted by blood transfusion and organ donation. It's not spread through coughing, sneezing, or touching. The incubation period is 3 to 14 days. 80% of the people are asymptomatic, with the symptoms ranging from fever, headache, tiredness, and body ache, and occasionally skin rash and swollen lymph nodes. Severe disease causes neck stiffness, stupor, disorientation, coma, tremor, convulsion, and paralysis. People over the age of 50 and immunocompromised are severely affected. Diagnosis is done by the IgM antibody capture ELISA in the early stages and RT-PCR. Virus isolation is done by cell culture, and IgG antibody is also used for late infection, to screen late infection. Prevention has been, the animals are vaccinated against the virus, horses, and the prevention is by controlling the mosquito transmission, reducing animal to human transmission during slaughter is of very importance, and due to transmission due to blood transfusion and organ transplant is also to be implemented. Zika virus has a sylvatic cycle and an urban cycle, and the sylvatic cycle is found between the chimpanzees, monkeys, and the mosquitoes. It spreads out to humans through the mosquitoes, and it's spread among humans using by mosquito. It's transmitted through sexual intercourse and blood transfusion as well. It is identified in the rhesus monkey in the tropical Zika forest in Uganda, and it's found widely in the African countries. There was a global outbreak in 2007, and there was an outbreak in 2015 as well, and there was a lot of people infected, especially in Brazil and Puerto Rico, Colombia, and Mexico. In India also, there was a few cases in Gujarat. 80% of the people are asymptomatic. Symptoms include mild fever, skin rash, conjunctivitis, muscle and joint pain. It causes retroorbital pain, anorexia, photophobia, GI disorder, and sweating and lymphadenopathy. Young kids are affected with a birth defect called microcephaly, where the brains are not developed at all. And neurological complication like Guillain-Barré syndrome is found in adults. Lab diagnosis is done by identification of the virus in RNA by RT-PCR, and antibody detection by ELISA or plate reduction PRNT. And vaccines under trial. Prevention is by personal protection, biological control by larvivorous fish, chemical control by organophosphate compounds, and yellow fever virus also has a sylvian cycle and an urban cycle. It's transmitted by mosquitoes again. The disease occurs mainly in South America, Central America, and Africa. It's not found in India. Reasons probably is because we are protected because of the cross-reactivity between dengue virus and this virus. And since dengue is endemic in India, we are all protected. The incubation period mostly is about a week. Incubation period is about a week with an acute phase where you have fever, headache, backache, muscle pain, and a severe phase where you have jaundice, dark urine, reduced amount of urine production, and bleeding from the gums, nose. Lab diagnosis is done by IgM ELISA, neutralization test, and molecular diagnosis by RT-PCR and isolation of the virus done by cell culture. Yellow fever vaccine, which is a live attenuated vaccine prepared in allantoic cavity of chick embryo, is given as a single dose, and it's effective within 7 days of administration. Prevention includes vaccine and avoidance of mosquito bites in areas where yellow fever is endemic. Ebola is one of the largest and deadliest outbreak virus, which is used as a biological warfare weapon as well. And it's found, it belongs to the family Filoviridae and the order Monogocaravels, non-infectious RNA genomes. It's found in fruit bats, and the fatality is about 50%. Transmission is by blood, body fluids, contaminated body fluids of people who are sick, and infected fruit bats. The virus can survive for a long time on surfaces. The transmission occurs through bat, and the incubation period is 2 to 21 days. Clinically, patient present with fever, severe headache, muscle pain, weakness, fatigue, diarrhea, vomiting, and lab diagnosis is by looking at the low white blood cells and platelets and elevated liver enzymes. The specimens are whole blood and serum. Ebola virus is a group 4 agent which requires high containment facility for working with it. Viral inactivation methods are chemical inactivation, gamma irradiation. Antibody capture ELISA is done for diagnosis. Antigen capture detection methods and serum neutralization tests are also done. RT-PCR is the molecular is done for molecular diagnosis of the virus. Electron microscopy and virus isolation by cell culture is also done. Then it has to be done in ESL2. Prevention is focused on the containment of the, mainly the nosocomial transmission has to be controlled and patients has to be isolated. Vaccine is also on trial. Marburg is a virus which belongs to the same category which causes Marburg hemorrhagic fever. This is again a category A bioterrorism agent, which was first recognized in 1967 with a case fatality of 50%. This transmission is by fruit bat and spreads among humans through human to human transmission, blood secretions and other body fluids. Diagnosis is based on clinically the patient presents with high fever, severe headache, and severe malaise. The incubation period is about 2 to 21 days. It causes watery diarrhea, and it causes the ghost-like drawn appearance in or drooped eye appearance in the patients with an expression like less face. And lab diagnosis is done by ELISA, RT-PCR, and also antigen capture method. Confirmatory method is the virus isolation by cell culture and serum neutralization test. There are no licensed vaccines again. Supportive care is given. The control is by reducing the risk of bat to human transmission and human to human transmission in the community, or especially nosocomial transmission. Lassa virus is another acute viral hemorrhagic illness with a of 2 to 21 days duration. It subsides on itself. It's caused by single-stranded RNA virus, which belongs to the RNA viridae. It's transmitted, the animal reservoir is the mastomys rat, and it's endemic in the African countries. It was first reported in 1969, and it's named after the town in Nigeria. Mode of transmission is by ingestion of contaminated food, inhalation of tiny particles, or air contaminated with the rodent feces, direct contact with urine droppings of virus through touching, and from human to human contact from exposure to virus in blood, tissue, secretions, and it's also causes high nosocomial transmission. Sexual transmission also has been reported. The incubation period is 21 days. Fever, general weakness and malaise are the sickness reported, with headache, sore throat. In severe cases, facial swelling happens, and shock, seizure, tremor, disorientation, and coma in laser. Hello? Hello? Diagnosis is done by RT-PCR and antibody ELISAs, antigen detection method, virus isolation by cell culture, and prevention is control, avoid contact with mastomus rodents and food, and keeping the house clean. Rift Valley fever is identified in 1931. The case fatality rate is very less, with 1%. The vectors are Culex and Aedes mosquitoes. Transmission is by blood, body fluids, and tissues of infected animals and bites of infected mosquitoes. There's no human-to-human transmission recorded. Diagnosis is done by by reverse transcriptase PCR, IgG and IgM ELISA, and virus isolation by cell culture. The incubation period is about 2 to 6 days. Prevention is basically by restricting the movement of livestock and reducing the risk of animal to human transmission from animal products, and personal and community care. Crimean Congo hemorrhagic fever, which was first characterized in Crimea and then in Congo, hence called Crimean Congo hemorrhagic fever. In India, the first outbreaks were reported in 2011 in Gujarat. 3 other outbreaks have been also reported in 2014 and 15. Case fatality is about 40%. The causative agent, which is the Crimean Congo hemorrhagic virus, which is a which belongs to Bunia viridae. The transmission happens using adult heart tick, and the natural reservoir is the heart tick and the wild animals like rats, camel, cattle, and sheep, goats. Animal to human transmission and human to human transmission also happens. It's transmitted through tick bite. The incubation period is 1 to 13 days. Clinically, the patients present with fever, sore throat, dizziness, neck pain, stiffness, backache, and headache. Patients might experience sharp mood swings and become confused and aggressive. The agitation is replaced by sleepiness and depression after 2 to 4 days. Mortality rate is about 50%. The second week of illness, and differential diagnosis has to be done with malaria and leptospirosis. The virus has to be handled as a group 4 virus. Sample collection is blood samples, serum, plasma, urine, and tissue samples. Molecular diagnosis is done by RT-PCR, and virus isolation is done during the early stages of illness. IgM and IgG ELISAs are done for diagnosis of virus. Prevention is by controlling the tick, by using protective clothing or applying repellents. No vaccines available yet. Swine flu is reported in India and with a case fatality of 5.3%. They are an enveloped RNA virus which belongs to Orthomyxoviridae family and has two surface antigens, which is the hemagglutinin and the neuraminidase, according to which they are called H1N1 or H2N2, depending on the types which are present. There are 18 neuraminidase and 12 hemagglutinin antigens, according to which they are named. The reservoirs are animal and bird. The seasonality is usually found around the monsoon. Diagnosis is done by, the incubation period is 1 to 7 days, with the patient presenting with headache, myalgia, cough, sore throat, fever, chills, and vomiting. And specimen is nasopharyngeal swab, nasal aspirate, or nasopharyngeo swabs. Bowel and sputum can also be used. And the virus, the specimens has to be transmitted in VTM at 4 degrees centigrade. RT-PCR and rapid influenza antigen tests are done, and immunofluorescence and virus culture can also be done. Prevention is by avoiding swine to human transmission and human to human transmission. Early detection and containment of the virus are the main methods by which it can be controlled. Personal protection measures and N95 masks are a standard protocol for controlling the disease. Zoonotic diseases in general can be controlled by real-time surveillance and reporting, rapid and accurate lab diagnosis, and early warning and rapid response to by public health authorities. Immunization of the host if the virus is already known, and vector control. These are the major methods by which zoonosis can be controlled. Thank you.
Thank you, Dr. Hubert. It was an excellent presentation, giving us a very large bird's eye view of all the zoonotic diseases that are prevalent and very important in the current scenario. We thank you. I have a few questions that have come up in the chat box. Would you like to answer, sir?
Sure.
Okay. So the first question is, what are the general symptoms of a zoonotic disease, irrespective of their origin? They've asked, what are the general symptoms of a zoonotic disease?
General symptoms would depend on the how it's transmitted. Let's say if it's a respiratory virus, then you would get cough, fever. The most common symptom would be fever and malaise, tiredness. Those are the common symptoms which you would see after any infection, I think.
Okay. Thank you, sir. Another question. Shall we consider COVID-19 as a zoonotic disease by origin?
Yes, it is. Yes, COVID-19 is a zoonotic disease, which because it's come from the bats. I didn't go over the coronavirus because there's a lot of talk going on. And since this is a big talk, I probably would not be able to complete it. So I skipped that.
Okay. One more question. Which organism acts as a reservoir to more viruses?
Reservoir, the main reservoirs are the cattle, because the animals which we have contact to are the major reservoirs for viruses which can come to humans. So the reservoirs for viruses which would cause human infections are the animals which are in close contact to humans.
Okay. One more question. Maybe we take it as a last question. Can COVID-19 transmit from the infected person to pets, pet animals?
I'm sorry, I didn't get the question.
Is there a possibility of COVID-19 to be transmitted from an infected human to their pets, pet animals?
To their pet animals, there are reports of the pet animals getting infected with COVID-19. But as it's too early to say, but I think there are reports of COVID-19 infecting the pets.
Okay. One more last question. What is the question? One second, I'm sorry, I could not go through. What is the best diagnostic kit for zoonotic disease?
For any infections, RTP, the PCR, molecular diagnosis is the best method because it's direct and it's now easily available. Then ELISAs, which are cheaper and it's easily doable at any place. Those are the two methods. IgM ELISA would be the first to come, and IgG would be the ELISA to be followed later.
Okay. Thank you, Dr. Hubert. It was a nice session. Most of the participants have appreciated and they have commented that it was indeed detailed, giving them excellent overview of the clinical presentation and the diagnostic features. Thank you very much for joining us in this session today. Thank you, Dr. Hubert.
Thank you, Dr. Mabel, for having me.
Without any further delay, let us move on to the next speaker of today's session. Today we have Dr. VJ Jins, who is here to be the resource person of the second session. To give a short introduction about him, Dr. VJ Jins did his BSc Zoology in Payyanur College, Kannur University, MSc Zoology in Madras Christian College, and PhD in Salim Ali Center for Ornithology and Natural History. His professional experience includes being a junior research fellow at Kerala State Biodiversity Board, research fellow, project fellow, senior research fellow at SACON, and now a research associate in the Department of Zoology, Sikkim University. He was awarded Sanjeeva Raj Young Research Award by MCC and Miriam Rothschild Internship by University of Cambridge, UK. He has published 7 research papers in peer-reviewed journals and has presented in 7 international conferences, including China and United Kingdom. His research includes people biodiversity register preparation, distribution of faunal groups in Agastya Malai Hills, ecological baseline assessment of Park Bay, cumulative impact assessment of hydroelectric projects in Sutlej River Basin, biodiversity components in landscapes of Darjeeling, Sikkim, Himalaya, discovering a new species of Europeltis, bird species richness and reptile richness along the Western Ghats. Dr. VJ Jins is at present the research associate in the Department of Zoology, Sikkim University, Gangtok. Dr. VJ Jins, we are glad that you are here for this session. Now I request Dr. Jins to begin his presentation. He may share his screen and the questions will be taken towards the end of the session.
Thank you, Mabel, for the introduction. Am I audible?
Yeah, you're audible.
Okay, thank you. And
you
I would
can
like to
present
thank.
your screen. Okay.
Yeah. Yes. Is it visible now?
Yeah, it's visible. You can share it in full mode, okay? Full screen mode. Yeah, good.
Done?
Yes.
Yeah, first of all, I would like to thank St. Xavier's College Zoology Department for inviting me as a resource person for this webinar series. And hope all of you had a nice session. And this will be a entirely different session for you. And I'll be talking about some part of my PhD research. So for ecologists and evolutionary biologists, the major question is why there are many species in some place compared to other place. So this is a major question for any ecologist. So for addressing that, ecologists take different steps. So here I will explain some of the aspect uh, which talk about mountains and species diversity patterns with a case study of reptiles from the Western Ghats. So why mountains are very important for studying ecological and species diversity patterns. As you know, mountains are very important, and it is considered as sacred places for in many societies. And from 19th century, scientists have identified there is a significant difference in species richness and habitats across latitude and altitude. Uh, Main reason is you can see different climatic conditions, the variation in temperature and precipitation. Once you go from lower altitude to higher altitude, or even if you're considering latitude, when you go from low latitude to higher latitude, there will be a uh, significant difference in temperature and both precipitation. As you can see in the figure, there is at least 0.5 to 0.7 degrees Celsius difference in temperature when you go from each 100-meter elevation or each 1 degree latitude. So there is a predictability in temperature and precipitation gradients. And if you consider the mountains compared to latitudes, mountains can be a natural laboratory, which is comparatively easier when compared to latitudes, because if you want to cover a few degrees in latitude, we have to cover a larger geographical area. At the same time, if you want to study gradients in environment, the mountain can act as a very important natural laboratory. So here, as you know, the latitudinal patterns are one of the most explored biodiversity patterns. Scientists have studied from long time back, and they have also proposed some major hypotheses to explain why there are many species. For example, as you see in the diagram, the tropics have higher number of species. The red color shows a higher number of species, and towards high latitude, you can see there are small number of species. So for explaining this pattern, scientists have proposed different hypotheses. So one of the hypotheses is called mid-domain effect. This may be some of you might have heard it, but for some of you it will be a new thing. So this is actually explaining the species richness patterns based on a geographical limit. Suppose there are two geographical limits, and the species are randomly distributed within the limit, then there will be a higher number of species in the middle of the limits. So this hypothesis is called mid-domain effect. And in same way, there is one more hypothesis called the Rappaport rule. It talks about geographical range size of a species. It explain in a way, range size is based on the climate, how a species can withstand with extreme climate. Based on that, the range size will be different. For example, species in occupy high extreme climate, they have a tolerable they are very much tolerable to extreme climate, and they may extend their range to extreme or different climatic situations. And once you come to elevational patterns, as I told before, you can consider elevational gradient or the altitude as a miniature form of latitude. So you can apply the same mid-domain effect, Rappaport's rule, these concepts in elevational gradient. That means across the altitude. So what a mid-domain effect predict is if there is a if the species richness is limited by a geographical boundary, then it will restrict within an lower and upper limit. So as you can see in the right side, there is a graph showing a lower limit with a green color and a upper limit with red color. So if species richness pattern follows mid-domain effect, the empirical richness or the experimental richness of species will fall within this upper and lower limit. And according to Rappaport rule, the higher elevation species, most of the time, higher elevation will have extreme climate. For example, the higher elevation will have extreme cold conditions. So the species, when extreme cold climate comes, the species may come down to lower elevation to adjust with the climate. So they are supposed to have a higher range size. And also when we consider a elevational species richness pattern, we have to consider 2-3 different diversity aspect. So the major aspect which we cover is the alpha diversity, which is the local biodiversity in a mountain. And if you have to address the beta diversity, you have to compare between two or more different elevational zones. Suppose we are covering from lower elevation to the peak of the mountain, we will be dividing into elevational zones, and the composition of species within this each zone will be different. So not just taking this richness, that is the number of species. In beta diversity, we also consider the composition of the species. In gamma diversity, we have to consider the entire region to account all the species in the area. And for explaining this species richness pattern, there are 4 major hypotheses. As I told before, some of the hypotheses, the mid-domain effect and Rappaport rule comes within the spatial hypothesis, and the Rappaport rule is also having a component of climate. So there are 4 major aspects. One is the climatic aspect, and another is spatial, and the third is biotic, and the fourth is historical. And like climatic factor include the precipitation and temperature and other derivatives of the climatic factors, and spatial factors include the area, which is there is a concept of species area relationship. It explains if there is larger area, it can hold a large number of species. And mid-domain effect I explained before. And the biotic factors include the species pool in a region. So the species richness of a particular elevation zone or a particular habitat is a subset of the larger species pool in that area, and also the habitat. So some habitat may be very complex and some habitat might be very simple or disturbed, and all these factors will influence the species richness in that area. And the historical or evolutionary factors include the historical events such as diversification of species and species dispersal and all these factors. So if you consider different studies on different taxa addressing the elevational patterns, you can see 4 major patterns. The first one is a decreasing pattern. When we go from lower elevation to higher elevation, there is a the species richness is decreasing. There is a monotonic decline, and the next pattern is a low plateau. That is, a lower elevation will have a plateau, and then it will decrease. And the next pattern is a like lower elevation will have a plateau and mid peak, and after a peak it will come down. And the fourth pattern is a mid elevation peak. So the mid elevation will have the higher number of species. So if you look at different taxa, you can see non-flying small mammals and plants. They mostly show a mid elevation peak. And if you look at reptiles, mostly you can see the decreasing pattern. So these are different studies. You can see last 1 decade there are a lot of improvement in studies along elevation gradient. So here I'll be talking about how elevational gradient influence reptile richness in some part of the southern Western Ghats. So why reptiles are a very important taxa to study different ecological patterns. So they are ectothermic in nature, means they are influenced by the ambient temperature, and they have very diverse life histories, and the ecology of different groups within reptiles. You can see very morphologically diverse and ecologically different species and groups, and they are compared to other taxa, for example birds and mammals, they are less mobile, so mostly they will have a small range size. And if you just take Western Ghats itself, there are many species yet to be described or explored. You can see every year there are at least 2-3 new species described from Western Ghats, and same way many other part of Indian subcontinent are yet to be explored for the reptile diversity. And you can see in the right side this Calliophis bedome is a coral snake, Bedome's coral snake, which was a new report to southern Western Ghats. It was the first report to Kerala. Like when I was doing a roadkill survey, I just observed the species. And this Europelis bupete was described in 2018. It is known only from Anekati Hills in Nilgiri Hills. And so if you take Western Ghats, it is a very important biographical region to study any biodiversity patterns. So it's a chain of mountains along west coast, and there is a elevation range up to 2,700 meter, and it's one of the hottest biodiversity hotspot of the world. And compared to Himalayas and other mountains, it is one of the older mountain range, and it is well known for its endemism. For example, more than or at least 50% of reptiles are endemic to the Western Ghats, and 70% or more than 70% of amphibians are endemic to the Western Ghats, and many of them are like the ecology and distribution of many species are not known so far. And we can also see diverse habitats, like starting from deciduous to evergreen patches, and also you can see a unique habitats called sholas, which is the high elevation. Mostly you can see this shola habitats above 1500 meter elevation. And as you know, Western Ghats is a fast changing landscape, as there are a lot of interventions in developments and other aspects. So we need to have a proper conservation planning for this landscape. And the climate change is also another aspect which we need to understand when we talk about species distribution patterns. When we come to the case study, so which I'll be talking about here, This was a part of my PhD research, which I did in Agasthyamala Hills in the southern Western Ghats, where I did the study on reptiles along elevation gradient. So the Agasthyamala Hills is known for its rich biodiversity and known for the medicinal plants and plant endemism, and it also have the indigenous communities called Kani tribes, and it is a well-known pilgrim center called Agastya Kudam, and every year thousands of people visit the area, and there are a lot of disturbances in the lower elevation due to pilgrimage and other tourism activities and human settlements. So this is a very important region, same time biodiversity rich region to address any ecological studies. And when you go from lower elevation to higher elevation, you can see diverse habitats, starting from moist deciduous in the lower elevation, and then semi-evergreen and evergreen patches. And in the top of the hill, you can see the patches of hilltop evergreen forest. So the diverse habitat give us an opportunity to test and compare different diversity patterns along elevation. And the method of my study was doing a reptile sampling and also accounting some of the habitat variables, and also collecting some climatic data. So the reptile sampling was conducted between March 2012 and December 2014. For 3 years, I've conducted a survey in the region, and the Agasthyamala Hills have elevation range starting from 50 meter to 1869 meter, but there are very less undisturbed forest below 100 meters. So I have considered elevation range starting from 100 meter, and systematic sampling using visual encounter survey was conducted, and dividing each the entire elevation zone into each 100-meter zones. This visual encounter survey consists of search by 2 people for 1 hour, and it includes turning different microhabitats, like we have to search in the leaf litter, turning the stones and scanning vegetation. And it's an intensive search and a standard methodology for reptile sampling. And for accounting habitat variables, mainly we considered vegetation density or tree density by taking 10 into 10 meter plots. So the tree density can be considered as a surrogate of habitat complexity, because when there is a high tree density, there will be more microhabitats for the reptiles, and so we expect that there is a more habitat complexity, there will be higher number of species. And climatic data, so it is difficult to collect on ground climatic data for a longer period of time. So we have used available climatic data from the CHELSA database, which is a fine scale data for a long term, like it starts from 1979 to 2013. And this is widely used for modeling species richness and ecological modeling. So this sampling effort in different elevation zones and the species observed in each elevation zones. During the sampling, I observed around 47 species, a total of 47 species in sampling. And if you look at the species richness along elevation, you can see when we consider different groups, all reptiles, I've taken all reptiles together, and also try to look at different groups. For example, snakes and lizards, they are different in their ecology and other habitat requirements. So I try to analyze snakes and lizards separately, and also endemics, Western Ghats endemics, to see whether they have any different patterns compared to uh, other groups. So this will also help us to understand the habitat association of each group separately as well. So you can see all reptiles, when we consider all reptiles together, there is a declining pattern of species richness from lower elevation to higher elevation. And same pattern you can see in lizard richness as well. But in case of snakes and endemic richness, there is no significant pattern observed. And similarly, for the mid-domain effect, if you consider this dotted line is the upper and lower limit, and you can see the species richness of all reptiles. The diagram, the first left diagram, shows the line in the thick black line is the species richness observed, and it declined to high elevation, and it does not fall within the upper and lower limit of the null model prediction. So this null model prediction is to just check whether it falls within within the concept of mid-domain effect. So lizards also follow the same pattern as all reptiles, but if you look at the snakes and also endemics, some portion of the empirical richness or the observed richness falls within the limit, but it also did not show any significant statistically significant pattern of mid-domain effect. So Rappaport rule, the test of Rappaport rule is conducted based on the relationship between range midpoint. Range midpoint is deciding whether a species is a higher elevation species or a lower elevation species. And according to Rappaport rule, higher elevation species, when the range midpoint is high, the range size of a species should be also high. But as you can see, it doesn't follow a pattern. If it follow Rappaport rule, it should, most of the species, each black dots are like the richness value of our species richness number of each elevation. If it follows Rappaport rule, it will be close to the red line. But here you can see it is not following Rappaport rule. So the second graph I'll explain in the next step, because for explaining the mechanisms for elevational pattern, we have done linear regressions to see what are the explanatory factors for each group. What is the factor explaining richness pattern along elevation? So along with other environmental factors, temperature and the area availability. The area is the available area in each elevation. Like in my sampling, I've considered each 100 meters. So the available area in square kilometer in each elevation zone is also considered for the analysis. So the Rappaport value is like, if we consider the Rappaport rule in the second plot, so it is adjusted to a particular elevation, and we can see how many species will be there if it follows Rappaport rule. So even though we didn't get a significant Rappaport rule, like the way of analyzing this elevation pattern includes the Rappaport rule and also the mid-domain effect values to along with other environmental factors. So here in case of reptiles, the best model showed a Rappaport rule, but when we analyzed separately the Rappaport rule, it was not showing a significant relationship. So the next model is having the temperature and having a significant association, and so the temperature turned out to be the best predictor variable for reptile richness. And the same pattern was observed in case of lizards also. The temperature was the best uh, explanatory factor. But in case of snakes and endemics, uh, there was no significant model uh, for explaining these patterns. And same time, if you look at the turnover along the elevational zones, uh, the turnover is nothing but the composition. So the species richness may be same in two different zones, but the composition may be entirely different. So if you look at the turnover pattern, we will come to know whether there are some elevation with a very unique set of species. So here you can see some of the higher elevation having the value 1 means the complete species composition is different. For example, you can see 1200 to 1300 meter zone is having a entirely different set of species when compared to 200 and 300. So in the same way, there are many higher elevation zone having a different set of species. So when we take the endemism within Agastimalai Hills, so I told that a total of 47 species I observed in the sampling. But apart from sampling, since reptiles are very rare to find during the sampling, other observations were also included for this analysis. A total of 70 species were observed from the region, and out of that, 44% of the species were endemic to the Western Ghats. It also matched with the entire Western Ghats endemism, which is also close to 45 to 50%. And if you see separately for lizards and snakes also, lizard endemism is very high in the Agastimalai Hills. About 55% is endemic to Western Ghats. These are some of the endemic species found in, these are the endemic Western Ghats endemics found in Agastimalai area. So I have given the elevational range which I observed, especially the first lizard species, the Calotus numericola. It is interesting that the species was found only between 500 to 700 meter during my survey. So similarly, there are many species which have a very small elevation range. So in that way, we also have to consider when we look at the species, we also have to consider their elevation range when we bring out a conservation plans. There are a few conservation concern species. These three species are endangered under IUCN categories. The Autocryptis beddomei is a very interesting lizard, which is also called kangaroo lizard. And you can see one turtle which I observed as a roadkill. This is an endangered species and very particular to low elevation evergreen habitat. And this was observed during a roadkill survey. It's a very important. It was a very important observation during my survey. And there are some other species like you can see Indotextual Travancore. It's a Travancore tortoise, and you can see the king cobra, which these two are vulnerable species. And these are some of the conclusions of my study is the temperature was found to be a major influencing factor for elevation richness of reptiles. And when we consider this temperature factor, it also have implication when we consider the climate change aspect, because if there is a change in temperature and other climatic variables, it will critically affect the distribution of many species, which are like some of the species are very range restricted and very very much associated to their microhabitat and other climatic variables. And there were high turnover between some of the elevation zones, but like I was not able to address the beta diversity uh, uh, in this study, because there were unequal sampling effort along elevation. So if we just address species richness pattern, that is fine. But once we have to address the beta diversity pattern in detail, we also have to take care of some of the sampling issues in any study. So by considering the turnover pattern, like I feel that there is a need of exploring beta diversity patterns across many of the hill ranges in Western Ghats, which will help us to understand the diversity in a better way, and it will help us to implement some of the conservation planning, and also like further explorations of elevational patterns and experiments are very important. As I told, it is one very important aspect which we can study the species diversity and explain the factors influencing these diversity patterns. And documenting elevation range size is very important when we prepare any conservation action and planning. So with this conclusion, I would like to end my presentation, and I think if there are some questions, I'll be happy to answer.
Dr. Jins, thank you for the nice, colorful presentation related to the reptiles. And thank you for explaining that temperature has played a major factor in influencing the distribution of reptiles. There's one question from the participant. In your study area, have
Yes.
you witnessed parasites influencing the distribution of reptiles?
Sorry, can you repeat?
In your study area, were you
Yeah.
able to find any parasites that was influencing the distribution of reptiles?
I haven't done any detailed study on the parasites. Like I have observed not in my study area in Agastya Malay, but in some other especially for the new species which we described, we have found some kind of infections for the snakes, like kind of some mouth infection, kind of bacterial infections. But as of now, I haven't observed any parasitic um, infection for the reptiles in my study, because it was mostly addressing the ecological and species distribution patterns.
Okay. One more question. If climate change has affected, whether climate change has affected the richness of the reptile species in Western Ghats?
This climate change aspect is a very complicated aspect to cover, because we first have to see for each species what is the tolerance of climate and what is the range they can there is a main stream of studies which emerging. It is called climate change vulnerability assessment studies. So in that we can model each and every species to see what will be the impact of climate change on each species, because for each species their tolerance to temperature or any other disturbance factor will be entirely different. So we have to have a detailed understanding of the ecology of different species. Then only we can talk about how the climate change will impact each species. For now, we know that reptiles and amphibians, they are very much dependent on the temperature and precipitation gradients. So if there is some alteration in temperature or precipitation, it will definitely affect both reptiles and amphibians.
Yeah. One more question. You have focused only on reptiles. So what are the other kind of animals that are expected to be found in sholas?
Actually, the shola habitats, which I was um, telling, is mostly found above 1500-meter elevation. There are many species like, for example, uh, some birds and some reptiles which are very specific to this habitat. But in Agastyamalai Hills, if you're taking, there are no proper shola habitats. In my study area, there was no proper shola habitat, but there are like hilltop evergreen forest because the maximum elevation in Agastyamalai is 1860. So, but if you go to Anomalais and other north part of the Western Ghats, you can see a larger shola um, habitats where you will find uh, more species which are very specific to the region. As of the Agastyamalai concerned, there are like there are no species which is very particular to this habitat, but some species are like some large scale pit viper, which is very particular to higher elevation in the region.
Okay.
So
Uh,
yeah.
okay, thank you. One more question. Did you see any change in the number of animals when you visited in different elevations and different seasons?
Yeah, actually, this definitely there is a difference in number of species across elevation that I have already presented here. There is a decline in species when we go from lower elevation to higher elevation, and covering different season is like for amphibians, it is very important to cover different season because mostly the activity is very particular to monsoon. But in my
Okay.
study, I was concentrating more on reptiles, which that also season is important. But I was trying to cover both dry and wet seasons and sampling different habitats so that I can um, record all the species in each elevation gradient covering different seasons and different habitats. So this is the main aim is to record all the species in a particular habitat covering different seasons. Definitely there are changes in their activity in different seasons.
Okay. There's one interesting question. How was your experience when you found a new Europeltis species?
It was the process actually took 2 3 years because uh, it was most common species in Sakon campus. Like I was observing the species for a few years, but it was already reported as a some other species which needed some more taxonomic work. Actually, when I got some species from my study area, Agastyamalai Hills, I just try to compare between different species, and then I discussed with some expert, and I got an opportunity to visit Natural History Museum, London, and then we actually in the process we realized that it could be a different species, not it was earlier reported as Europellis elioti, a different species. And in this process, it was very surprising for us to know that one of the most common species in that campus is going to be a different species.
Okay.
So it was quite surprising.
Okay, one more last question we'll take. What are the favoring conditions that you think for the effective diversity of reptiles in the Western Ghats?
Effective condition, you're asking?
Yeah, what are the favoring conditions for the reptile richness in a particular
So
area that is in Western Ghats?
that is a very difficult question to answer because each species, for uh, reptiles and amphibians, especially reptiles, if you consider different species, they need a very, not just uh, environmental fact, means not just climatic factors, even the microhabitat factors are very important. Suppose there is a favorable climatic condition, but the habitat is disturbed, then the species will not have a proper microhabitat to uh, uh, like use. So in that case, there should be a undisturbed habitat, and also like a uh, temperature which is like, they are already adjusted with a particular set of climatic factors. So if the combination of different factors will actually influence their uh, observation. Like if you have to have a combination of different factors, including climate and habitat, this will favor their existence.
Okay. Can you hear me?
Yes, I can hear.
Okay, okay. So the separate, a repeated question just like that. Other than temperature, are there any other influential features for the distribution of reptiles?
Uh, in my analysis, I found temperature as a major factor, and since the disturbance factor, no, like this is a pilgrim area, so if we can also address the disturbance factor, it will be also an important factor to cover. But in my study, I haven't addressed that factor because my major idea was to look at the species richness pattern, and taking like, if you want to cover the disturbance gradient, that needs a different kind of approach. So I was trying to address only the richness pattern, taking only undisturbed habitat. So I feel that if we can also address the disturbance factor and in the area, so it will be a interesting factor to address.
Thank you, Dr. Jins, for your wonderful presentation, and you have answered almost all the questions that was posted by the participants. We thank you for gracing this occasion with your virtual presence. So, on behalf of the organizing team, I would like to extend thanks to the resource persons of today's session. We will post the feedback link right now in the chat box. You can fill it up, and please ensure proper submission of the feedback forms. Only then you will be receiving the certificate.
Thank you, Mabel. Thank you for
Now
inviting me.
I request okay, okay. Thank you, Dr. Jins. Now I request our one of the organizing secretaries, Dr. Elizabeth, to propose the formal vote of thanks. Good afternoon, all. Sorry. Good afternoon, all. As all good things come to end in life. On behalf of the management, I take this opportunity to propose vote of thanks to those who have directly and indirectly contributed to this webinar on national level Zoology Webinar Series 2020, organized by our institution. At the outset, I thank our resource person, Dr. Hubert and Dr. V. Jins We are really enlightened with your knowledge and presence. We are thankful to our respected Reverend Father Rector, respected. We are thankful to our respected Reverend Father Rector, Reverend Father Secretary for the motivation, and I would like to thank our respected Reverend Father Principal for his enthusiastic support. A special thanks to the organizing committee, teaching and non-teaching staff for their unflinching support and coordination. Our heartfelt thanks to our participants for your cordial cooperation. With these warm words and a kind message, we move to the end of today's webinar. Thank you. Kindly join the meeting tomorrow at 9:30 a.m. Have a nice day. 9:30. Tomorrow at 9:45 a.m. Thank you. Thank you. வீடியோ மட்டும் போட்டேன்.